So as I said, the local ancestry are the genomic segments of an individual genome that can be traced back to an ancestor from a specific population living at a place and a time. So from this example, we can see there are two individuals. They have three markers at different genome location, which can be traced to three different ancestral populations. So in order to get the local ancestry segments, so there's a computational method named the local ancestry inference, or someone called deconvolution, which is a method to determine the ancestral origin of genomic segments of an individual genome, and particularly used for the mixed populations. So the inference process usually involving a reference population panel, which serves as a proxy of ancestral population. And during the inference, we are mainly trying to find the similar haplotype segments between the mixed haplotype query and the haplotypes in the reference population panel. And then we just copy the ancestral population labels from the panel to the queries, which can be inferred as a chromosome painting. So the local ancestry inference has many applications. So one of them is a genome-wide association study on the mixed population. Take an example of a tractor, which they incorporate the local ancestry cost in their association study model to show that their model can gain more power than the standard association study model. There has been many local inference methods developed in the past years, can mainly divide into two categories, the site-based and the window-based. So for the site-based method, they are mainly depending on using the Lee and Stephen hidden Markov model. The Lee and Stephen model says the mixture haplotype is a mosaic of two or more than two parental or ancestral haplotypes. So Recomb-Mix is one of the earliest method which used the Lee and Stephen model for the inference. And then later, Lothar formulated this problem as a graph optimization problem and used dynamic programming to solve it. Flare is another hidden Markov model-based method which incorporates many computational techniques for efficiency. And one of the recent work of a site-based method is sparse painter. Yeah.
Is not turned on. Nope, it is there. You're good. Sorry for the interruption.
Okay, no worry. So sparse painter incorporates the matches between the correct haplotype and the panels within their hidden Markov model. So another category is window-based. So mainly their machine learning-based approach. Recomb-Mix is one of the most popular local inference methods. It used the random forest classification to make the inference. Genomics use two different like stages to make the inference. They use support vector machine as to make their I guess initial estimation, and then they use the gradient boosting model to further smooth out the result to improve the accuracy. CLNet is another machine learning-based utilize the neural network, and Orchestra is most recent work which utilize the deep learning model into their ancestry inference method. Although there are many methods has been developed, there are still some challenges existing in the methods. I listed a few of them. So one of them is many methods, they require complex biological parameters input. So such as like recombination rates, mutation rates, the number of generations inside the mixture, sometimes can be hard for the user to provide those information for those methods. Another one would be high computation cost, such as long runtime, high memory demands, which can be some issue, when we scale this method to large volume or high resolution data sets. And the methods tends to not have those comprehensive evaluation, which the user may having hard time to pick which tools they want to use for their specific studies. So here we present Recomb-Mix, which is a site-based method. It's a approximation of a Lee-Stephen model. Particularly, we do not use any penalty for path switch within any population, and we use the normalized recombination rates for path switch between populations. We also use panel compression at haplotype level to gain efficiency. So here's an example of how we do the reference panel compression. So we convert the reference panel into a population graph following Lotus formulation. So each row represents a haplotype, and each column represents a site or marker. And between two sites, it's a fully connected graph. Then we further convert into a compact population graph by collapsing the nodes, which has the distinct allele values as well as the ancestral population labels. So using the compact population graph, we can reduce the search space, but also we preserve the ancestral signals from the graph by using the discrete ancestral informative markers, which shows on the green arrows here. So the discrete ancestral informative marker, or DAMs, we define them as the markers have allele values presenting in the panel of one population, but not in at least one another population. So we show that in the real world data, we plotted this DM density, which we did 7 populations in the Human Genome Diversity Project Chromes 18, and we can see that the DMs are densely available across the genome, which can be used for acquire the ancestral signal in our compact population graph. So then we use the dynamic approach programming to solve the this problem, finding the optimal path with minimum panel score. So here's our objective function. So mainly it composes of the mismatch penalty and the template change penalty. So they're similar to the imaging probability and the transition probability in the hidden marker model, also the loss function and the regularization function in the machine learning approach. So the major difference is for the template change penalty, we only applied the penalty when the path switch between populations, but not within populations. So here's an example of how we actually calculate the score and find the optimal path. So the score is calculated by adding up the minimum score of node from the last site plus the mismatch panel score, and then plus the template change penalty score. So we start from node S, and for each site, we would calculate the minimum penalty score for each node, and then we just go all the way to the end of the nodes, and then we trace back by selecting the nodes which has the minimal penalty score, all the way back to the starting nodes, and then we have this optimal path. Next, we just simply copy the ancestral population label from the nodes on the path to the mixed haplotype query. So this operation takes the number of markers times the number of populations in the reference panel, which is quite efficient, and it's independent from the number of haplotypes in the reference panel. Then we did the benchmarkers on our methods. So for the simulation part, so we use the real data, which is the Wensong Genome Project and the Human Genome Diversity Project as the founder population. So we feed them into the SLIM, which is a forwarded simulator. And then we run different number of generations and generate different size of reference populations. We also look into different the mixture cases. There are 3-way intercontinental, 7-way intercontinental, and also 3-way intercontinental. And for the performance evaluation, we use the squared Pearson correlation coefficient, whose definition is we follow previous published work. So here we show the three-way intercontinental case results. So we compared our methods to FLAIR, Genomics, Lotter, RFMix, and CellNet. So we can see for the three-way intercontinental case, the Recomb-Mix outperforms all other methods. And for the seven-way intercontinental cases, so Recomb-Mix still did a pretty good job, except the 200 generation case, which on the most right side. In that case, FLAIR and the Genomics did a pretty good on the distant generation case. Then similarly on the three-way intercontinental cases, Recomb-Mix did a pretty good job, except the 200 generation case. Then we show the runtime and the memory of our method. So we average the runtime for each hypertype query per each cases. So Recomb-Mix shows it's very fast, can get a one query done in like 1.4 seconds for three-way and maybe 4.3 seconds on seven-way, and also use the least memory among all the methods. Then we did some ablation study of our the properties of our methods. So the first one is the zero penalty for path switch within population. We can see that there's a significant improve of the accuracy of our methods if we do not give any penalty for the path switch within population, which is the green line to the orange line. Then we look into the template change penalty using the normalized recombination rates, which also further improves the accuracy, which is from the orange line to the blue line. Next, we look into the cases of the reference panel, because most methods they are heavily rely on the quality of the reference panels. Two of them, the issues we looked into are the imperfect panel and the uneven panel. The imperfect panel sometimes refers to misspecification cases, which means some haplotypes in the reference panel has a small fraction of the mixed segments in there. And then for the uneven panel referred as the underrepresentation, which means some population has more or less number of individuals than others in the reference panel. So for the imperfect panel cases, we mix the two populations, like 5 generations from the start of simulation, and then run the same setting. So we can see the Recomb-Mix performs pretty good, except the 200 generation case. So we also noticed that RF-Mix was very good in the most recent generation case, which is 15. For the uneven panel cases, so we use a different number of individuals of populations for both the founders population in the simulation, also generating a different number of populations for the reference panel. So in this case, all methods has some impact, but they're not significant. And also Recomb-Mix performs best in this case. To conclude my presentation, so we talk about the local ancestry, local ancestry inference, and the methods briefly, and also we introduced the Recomb-Mix, which use the zero penalty for path switch within population, and the normalized recombination for the path switch between populations. We also use the panel compression at haplotype level to gain efficiency, and we also our methods does not require complex parameters, and also we show that it's competitive with other state-of-the-art local ancestry inference methods. For the future work, we tend to try to use population-specific recombination rates to see if we can further improve the accuracy. I want to thank you to Dr. Shaojie Zhang, Dr. De Guizhi, and Dr. Asang Sanella for their contribution and the suggestions. And also I want to thank you for the grants from the NIH. So yeah, thanks.
Thank you. Yeah, we have a couple of minutes for questions.
Great talk. So I wonder what kind of input data we need for your software. Yeah.
The input data
out.
would be genotype-based data, and it can be
So
like,
WGS data or GYS data or WES data also will be okay.
right now, I guess our tool only takes the genotype, like whole genome sequence data in like a VCF format.
So it's a whole genome sequence data. Can we use whole exon sequence data is okay?
Not really at this time, no.
GYS data is okay or no? Genome-wide association status data is okay or no? Or not?
I think if it includes genotyping data, yeah, it is okay. So
I see. So you only can only input that whole genome sequence data is correct.
yeah, and also
is your tool available? People can use.
Yeah, it's available on the
Okay, thank you.
The GitHub. So yeah, and also it also requires the ancestry population label of those. Yeah.
Maybe one more question. Oh.
So really great talk. I'm looking forward to reading the proceedings, but I'm curious, with that representation, you said it needs whole genome data, but couldn't you do it at a much smaller scale as long as you had a long contiguous region with haplotypes? Would that be a problem, or
we can use like smaller like size of data. So yeah.
Okay.
So I guess the whole genome sequence there would be more appropriate because normally they contain more rare variants or ultra rare variants, which gives us like more strong like ancestral signal. So
how important are the rare variants? Sorry, if
maybe
yeah.
maybe 0.5% or 1%. I'm not sure the exact numbers, but yeah.
Okay. Now it's time to move on to the next speaker, and let's give a big hand